Hello, my name is Vanessa Raab. I'm a current junior at Drew University. I am pursuing my Bachelor's of Science in Biology, as well as two minors, one in Public Health and the other in Biochemistry. So today I want to talk about the research I've been able to do at Drew. So for me, a lot of it is DNA sequencing, but my main focus is working with microbiology. I've really found a passion for the field within my time at Drew. I started out as a neuroscience major, but then found out like bacteria in our gut does some really cool things for our body, and I became very interested in it in my freshman year. I worked with Dr. Neil Connors in the RISE program to help do literary research during my freshman year, and from there I started working with him during the summer for DSSI, which is the Drew Summer Science Institute. DSSI is a paid internship at Drew. For me, it was 8 weeks and I could work full-time in the lab, and I absolutely loved it and decided that's what I want to do when I'm older. That's what I want to do as a career, is to work in research. Um, so RISE is the Research Institute of Scientists and Meritire. Essentially, scientists from all across the STEM fields, uh, after they spend some time in industry, can come to RISE and become mentors for the students at Drew. This is a fantastic opportunity at Drew. Not only can students make their own research and do research as undergrads, but they're also receiving mentorship from either professors or from retired scientists that have been in the industry for many years, like in RISE. I've worked with two mentors within the RISE program. The first was Dr. Neil Connors, who's was my mentor during this summer, the summer after my sophomore year, which is where I conducted this research, and I've gotten these results that I'll be presenting today. From there, I've also worked with Dr. John Perkins, who I am now working with currently, to finish up this research project, as well as pursuing new parts of the research project, mainly mutagenesis. But to begin, my research project does have a formal title called DNA Sequencing of the Ribosomal Protein Gene, RPSL, in the strains of Kibdelosporangium species that produced increased levels of medically important antibiotic Kibdelomycin. I'll explain what this means. I don't expect people to know right off the bat what the heck I'm talking about, and so I'll go into that with some introduction. Okay, so Kibdelomycin is the natural product of Kibdelosporangium. Kibdelomycin in particular is an antibiotic. So it's really important because it's very important to look at new antibiotic candidates, especially as we're seeing more and more antibiotic resistant strains that are now becoming untreatable in modern times. So Kibdelosporangium is a bacteria that was discovered by Merck in the early 2000s. And when Merck is a pharmaceutical company, if you're not familiar with it, and from there they discovered that it's producing Kibdelomycin. The reason that they did not keep pursuing this project is because Kibdelomycin has poor pharmacokinetics. Pharmacokinetics is related to how well that drug is going to break down the body, how well is it going to actually work when it's being used in a medical condition. So what they found was Kibdelomycin was binding to different parts than they wanted to. It wasn't going to work as an oral antibiotic, but it's still a great antibiotic candidate, and potentially at Drew, we are focusing research on trying to change those pharmacokinetics so that they will work better and we can have good pharmacokinetics by chemically modifying the Kibdelomycin structure. So this is actually work that's going on with Dr. Golo's lab. My friend Ben's working with that. I, on the other hand, am working more with the Kibdelomycin production side of it, where my group is trying to improve Kibdelomycin production and fermentation of the Kibdelosporangium. So essentially, we're trying to figure out different ways to grow Kibdelosporangium, how to alter it so that we can get even more Kibdelomycin production, which would be better for manufacturing in a pharmaceutical standpoint. So ways that this strain was improved before I even came to Drew was done by Ryan Callahan. So she was working on a project based on the idea of adding antibiotic resistance to increase natural product production. So this is actually a well-used technique in pharmaceuticals. It's seen that in the Actinomyces family of bacteria, which Kibdelosporangium is part of, there is increased natural product production when you add antibiotic resistance. So with that, they've also looked at why that might be happening. And the reason that that might occur is because another group found that in ribosomal genes, particularly RPSL and RPOB, there was found to be mutations in those ribosomal genes, and they think that is correlated with increased natural product production. So what we've done, in particular Ryan Callahan, who was a student that came before me for this project, she started working with the wild type, which is F109. From there, it became streptomycin resistant. She worked through that and got the F strain. From there, she took the F strain that was streptomycin resistant and then made it gentamicin resistant, which gave F16. So with each time that this antibiotic resistance was added, if you see on the right side on the bottom, they have the amount of Kibdelomycin that was being produced by those strains on their optimal days. So the wild type produces a decent amount. You see an increased amount with that streptomycin resistant strain, and an even larger amount of Kibdelomycin production with that streptomycin gentamicin resistant strain. And this is really interesting to see that occur, and also to see that there is that increase in Kibdelomycin production. So with that natural product production, we're trying to see why might that be happening due to this resistance and to kind of explain it. And so that's where my project comes in. So I am working on sequencing those ribosomal genes I mentioned before, RPOB and RPSL, to see if there's any mutations that might explain why there's this increased natural product production. Over the summer, I've successfully sequenced RPSL with Dr. Connors, and currently I am working through sequencing RPOB with Dr. Perkins. So the way that I did this was I did not find any mutations at the end of it, but I'll talk through the process on how I got here. So to start out with, in order to isolate out the gene I want from each of my strains, I need to make primers. So essentially little tags are going on the ends of my gene. So I'm bordering it, and then this signals for machinery within the cell to say, cut here and amplify here. And so it'll cut out that part and start to amplify it. And so you end up, so this happens through a process called polymerase. We use a PCR, polymerase chain reaction. And so that will cut out that gene, and it'll just keep making tons and tons and tons of it. And we end up with this purified sample of our one gene. From there, I had to try out some different techniques to make sure I'm getting a very purified and isolated sample, so that it could be properly sequenced. So on the right side of the screen is actually some of the raw data I've gotten from our sequencer. We do have a sequencer in-house, which is our Thermo Fisher Seq Studio. We use it in the RISE department, and I'm very lucky to have that opportunity to use those kind of instruments hands-on. So from there, I've done Sanger sequencing, which is a type of sequencing I've now learned. I've learned quite a few techniques with at this point. So the raw data is shown on the right side. All the blue part on the top is all clean sequence. That's very much the machine is pretty sure that that is the correct sequence. Below that is how the machine is reading those nucleotides as it's sequencing, which has to do with the light and reflection of colors that are actually coming off the nucleotides as it builds onto the gene to try to identify it. It's a very cool process. I recommend for people to go watch more videos on how that works. It's very cool, and I don't have as much time as I'd like to explain it. From there, I took those raw sequences and I'm able to compare them through this Clustal. I use a Clustal program to present identity matrix, which essentially just compares every single nucleotide within that gene to see if there's any changes. You're hoping to see a point mutation, so just one nucleotide change at the end of the gene. We did not see that. There's a 100% match of all the genes of all three strains, whether it's the wild type, the streptomycin resistant, or the streptomycin gentamicin resistant. So with that, we found that there is no change in the RPSL. So with the future of my research, I'm currently sequencing RPOB. It's much more of a hassle because it's 3,000 base pairs instead of this 300 one. So 10 times the size. I've had sequencing problems because my Sanger sequencing techniques needed to be changed a little bit in order to accommodate for the size, as well as I'm working through a gentamicin mutation. I'm working through mutagenesis and chemical mutagenesis of the streptomycin strains to try to see if we can create new rifampin resistance. Rifampin is another antibiotic, so we're hoping that by creating that rifampin resistance to our already made strains, that we can keep increasing that Kibdelomycin production. So that's where the future of my research is going. Hopefully I'll be doing that over the summer as well as next semester. Currently it's on hold. If anyone has any questions, whether you're a current student at Drew, if you are a prospective student or someone who's just interested in this topic or wants to talk about STEM stuff or microbes or anything of the sort, I'm all for it. You can email me at vraab, V-R-A-A-B, B as in boy, at drew.edu. That's my email for Drew. I respond to it quite often because I'm on my email a little too much, but if anyone has any questions, thank you for seeing my presentation and enjoy your day of scholars. Bye.